Hey everyone, welcome to NIIMBL's Viral Vector Manufacturing and Analytics webinar. We're going to go ahead and get started. Can you go to the next slide, please, Dan? Great. We have just a couple of announcements first. Some webinar details is that you will be muted throughout the presentation. Please use the Q&A to ask a question or post a comment. We will be taking questions after the presentation, so please feel free to go ahead during the presentation and start asking questions so I can start collecting them. As a reminder, this webinar is open to NIIMBL members and non-members, so please don't post any confidential or proprietary information in the chat. There'll be a survey at the end of the webinar that will come via email. Once you complete the survey, you'll get immediate access to the webinar and the recording of the slides. Next slide, please. So again, welcome. My name is Angie Snell-Bennett. I am the program manager for the Viral Vector program. For those of you that were with us last year, this agenda may look like it has the same information. We do have some new technical updates to share, as well as Tim will be sharing some of the obstacles that we have faced over the last year. And we think that you will find these valuable, but we have a mix of stakeholders on the call, and we just want to make sure that we bring everyone along with us in this webinar. So Chris Yoakum is going to give a NIIMBL overview. Tim is going to give a program overview. And then Eric is going to give a technical overview and talk about how you can get involved in our program. And then I will come back with some wrap up, and that's where we will start taking questions. So next slide. All right, Chris Yoakum is our Director of Business Development, and he's going to give us a NIIMBL overview. Chris?
Great, thank you, Angie. And I'd like to thank Tim and Eric also for the invitation to be here. I appreciate that probably most of the people on the webinar are NIIMBL members, but it's always good to have a little bit of update. But there are also quite a number of non-NIIMBL members participating in the webinar, and we're really happy that we can share this information to both members and non-members to help people understand the important work that's taking place and the novel programs that are being added. We're very excited to be launching the Viral Vector program under Tim and Eric's leadership. Dan, next slide. So again, NIIMBL's mission and vision are stated here. We are a consortium of industry, large and small members, and academic institutions, and a few nonprofits working to accelerate biomanufacturing innovation. This was really driven home as an important opportunity as a result of the pandemic. That let the entire world know that we need to improve our manufacturing capabilities and capacities to meet the needs of the future. We are gathering our members so that we can lead the transformation and the development and adoption of the next generation technologies. And the Viral Vector program is a really great example of that visionary and aspirational goal to improve an important aspect of biomanufacturing, which all of our large companies agree is needed. And I'll be talking a little bit more about the members in a moment. Next slide. So for those of you who are not as familiar with what NIIMBL does, this slide covers a few of our mandates and probably a couple of others that I'll include. But it's to improve the competitive stance of the work being done in this country around de-risking manufacturing technologies that can improve the efficiencies of our capabilities and ability to respond to challenges. What's really unique, though, is bringing together people with different backgrounds and different expertises to share their practices and to promote regulatory harmonization, enhancing the ability, the safety, potency, and efficacy of manufacturing biopharmaceuticals. And lastly, one of the most important things that we've achieved already is to support this best-in-class education to build a workforce development capability for the future. I think there are few industries that have such challenge in meeting the current demands as well as the future demands for capacity and for capacity in manufacturing, as well as for a well-trained workforce to work in good manufacturing facilities. Next, Dan. NIIMBL is just one of 17 Manufacturing USA networks. This slide shows the others that are taking place. The concept is very simple, and that is we bring together large industry, small, innovative platform technology companies, and of course, the intellectual problem-solving capacity of our top-tier academic engineering institutions, all aimed at solving the problems and the bottlenecks associated with biomanufacturing. So this has been an experiment, I think, that has been extremely successful. As you see that most of these institutions have been around for a few years and continue to grow and to make an impact in their various technical R&D areas. Dan? I'll spend just a moment to dive a little bit deeper into what makes NIIMBL Consortium so unique. As I mentioned, we are a gathering place for subject matter experts from a variety of technical backgrounds, as well as the institutions which they're associated with. And it's NIIMBL's mission to bring these folks together, identify the bottlenecks and the challenges in biomanufacturing, and then help facilitate team building and putting groups together that can approach and solve those problems. So we have contributions from our large industry manufacturers. We have over 14 of the major global pharmaceutical companies part of NIIMBL. We have over 70 small to medium platform technology companies and a few pure play biotechs. And a couple of years ago, we instituted a membership level that would attract the startup companies, the university spinouts, so that they could be part of the ecosystem. And again, on the academic side, we have top tier engineering institutions from around the country. I think it's close to 35 at present. We have a large number of community colleges that are focused primarily on workforce development, but also doing technical innovation. And then we have access to our national labs, and that includes both FDA and NIST scientists from the federal government. We have participation from several state and local governments, as well as several economic development organizations. And that's how we're able to catalyze a number of various stakeholders to drive progress with our various programs and projects. And the last slide, I think, Dan? Well, two more. We've been very successful in our appreciation to the federal officials that support NIIMBL. Our Institute Director, Kelvin Lee, has been very successful at securing additional federal funding to support our efforts. And of course, a part of the funding that we use to cover the overhead and the operating costs of NIIMBL comes from the membership dues, which all of our small and large companies contribute to the success and to the financial stability of our consortium. And we're very grateful to all the organizations because we know what a challenge it is to justify a membership fee in any ancillary activity these days. So thank you to everyone for being a member of NIIMBL and contributing to our stability. And I think the last slide is just our membership slide, Dan. Yes. So here you'll see the logos of our large institutions that are members in the center there. As I mentioned, 14 of the major global biopharmaceutical companies. Our newest member, Boehringer Ingelheim, is there, and we welcome them aboard. At the top, again, the logos of some of the biotechs and the platform technology companies that have the innovation that we're trying to de-risk and demonstrate scalability and applicability to various manufacturing processes. Again, on the right and the left, we have the academic engineering institutions, and on the bottom, the logos of many of our nonprofits and community colleges. Again, we've been very successful, and I appreciate the effort of all the members of NIIMBL in helping spread the word, especially our members who talk to their peers about the value and the benefits of being part of NIIMBL. And for anyone who is interested in becoming a member, please feel free to reach out to me via our website or the connections that you have to this webinar today. And I think that's about it for membership discussion. Again, we are focused on identifying the needs, how NIIMBL can help meet those needs in biomanufacturing, and the outcomes and the impact. And if anybody would like a copy of these slides, just let us know. Thank you for the invitation to be here, and thank you for taking time out of your busy day to join us. And now I'm very pleased to introduce one of our NIIMBL fellows, one of our first NIIMBL fellows, Tim Charlebois. Tim spent his career at Pfizer, and we are very excited to have him, along with Eric, to lead this NIIMBL-led program in viral vector manufacturing and analytics. Tim, over to you.
Thanks very much, Chris, and thanks everyone. I agree with Angie that for those that have been closely following our program, there is not a ton of new content here. Hopefully, we will be able to articulate where we are and where we're going a bit more clearly. We officially launched viral vector manufacturing and analytics as a new NIIMBL-led program last month. And we get a lot of questions about what that actually means and how it relates to what NIIMBL's trying to do and how our members can be involved and contribute to the program and the overall mission to advance AAV-based gene therapy. So today, in addition to discussing what we plan to do technically, which Eric's going to go through, we wanted to take some time to explain what a NIIMBL-led program actually is and how it relates overall. I'm just going to go through that a bit, and then that will talk a bit about how we structure NIIMBL-led programs and how that relates to the opportunity to participate and contribute to our overall mission in the program. So next slide, please, Dan. So what are NIIMBL-led programs? This is really a generic slide, but it certainly applies to the viral vector program. The idea is to accelerate progress around strategic technical themes. So in our case, viral vectors, and we decided for the need to focus, we're going to focus on AAV-based viral vectors, at least initially. And to benefit the relevant biopharmaceutical manufacturing community. And to emphasize that we're looking to solve problems that are difficult or impossible to solve individually. And by taking a strategic approach, we can link technologies together into platforms. And so the notion is to try to take a longer view over a specific area, identify and recognize technologies that can be synthesized and delivered to the community in a pre-competitive manner that can be beneficial broadly and enable many to save time, effort, and money in a particular area. And additionally, by using the industrial-led consortium model, we can take a very interdisciplinary and cross-organizational approach. And that's very important because, as I'll talk about, we need to inform our intentions in a strategic manner, but we plan to make outreach to the broad community through requests for information, requests for applications, requests for proposals, and we want to drive that from this strategic and long view approach out into the community to drive activity. Next slide, please, Dan. So how do NIIMBL-led programs differ from the traditional project call program that you may be familiar with? So when NIIMBL launched in 2017, as Chris mentioned, we had membership and federal monies to distribute, and we created the project call program with an open call approach as a way to identify, relate needs to opportunities, basically. And so we would go through broad solicitations that were essentially driven by investigators. So as you can see, investigators submit specific proposals. So these are investigator-driven proposals that are responses to the project call program, and they're broadly solicited. They're not coordinated. So what we have is many dozens. I don't know how many projects NIIMBL has funded and run over the last 6 years, but it's a lot. Each is somewhat individual and standalone, and they aren't synthesized in a strategic manner. So that the notion of NIIMBL-led programs was to complement that the project call process with strategic thematic areas. However, again, we hope to do projects that are of similar duration to those that would come in the project call program. So we don't try to run something that's a single project for many years. We try to take discrete coordinated efforts, but in discrete packets of work that can be brought together and synthesized into something larger. So the definition of how, rather than defining a project by the submission of a proposal, we're actually starting with program participants in terms of defining what we wish to do over this time frame and then how we wish to do it. And so the definition, where the definition comes from is centered around the NIIMBL-led program. I'll talk about that a bit in the next slide, please, Dan. So how do we structure a NIIMBL-led program? So from the NIIMBL side, we provide program leadership. In this case, it's myself and Eric Hacherl who will be speaking next, and Angie, who opened the program, as a scientific program manager. And our role is to actually work with the community to define and then administer the program. And so we do this through a steering committee whose role is to provide strategic guidance and leadership to the program. Where are we going and how should we go? How will we go there? How is that going to contribute to the community overall? And then we have defined work streams, and as you'll see in our case, we'll have a work stream in viral vectors that's focused on the process area and one that's focused on the analytical area. But this is where the technical program gets driven from. So we will form a leadership team in the analytical space and in the process space that will essentially be converting the vision of the program into what work will the program do and how will it do it. And then this will then lead to the formation and funding and coordination of projects. And this is where the actual technical work is done. And I mentioned at the bottom that this NIIMBL-led program is primarily a technical program within the overall NIIMBL technical space, but we recognize that it's essential to integrate workforce capabilities with technology advancements in order to ensure that we are able to take advantage of the technology advancements, adopt, implement, and fully leverage them. So it's really important for us to not wall off workforce development as something that's its own thing and completely separate. We need to recognize that the capabilities of the workforce to use technologies is supported as well and integrated into the program. So this is a basic structure, and I wanted to emphasize that the work is conducted at the project level, and we'll talk about that more in future slides. But that we basically provide guidance and assembling a steering committee, and then technical direction through the workstream leadership, leading to the formation and execution of projects. And hopefully we can do that in a manner that's effectively strategic and contributes and maximizes the use of the overall resources that NIIMBL has available to us and the connections. Next slide, please, Dan. So I needed to talk about this slide a bit just because it's an important recognition of how NIIMBL is structured and the relationship of the membership tiers and benefits to the NIIMBL-led programs. So within the bylaws, the benefit to participate in the NIIMBL-led programs is defined. And as you can see here, the tier 1 industry members, which are the large companies that are at the high, have contributed at and participate at the highest level within NIIMBL have no limit on the number of programs in which they're able to participate fully. They have full IP rights to anything that takes place in the programs. They can contribute, they have rights to participate in the steering committees and in the workstream leadership of all of the NIIMBL-led programs. So that would include the viral vector programs. As we move down to the next tier levels among the large industry, the tier 2 industry have those same full rights to 2 NIIMBL-led programs, the affiliate silvers to 1 NIIMBL-led program, and the bronze to a single NIIMBL-led program work stream. So this is basically where the entitlement to participate in the programs fully resides and aligns with the tier structure, membership tier structure. But the key thing to then recognize, and this links to what I said in the last slide, is that the small companies and the academics and others are eligible to participate in specific program projects. So this is something that is open to all NIIMBL members. And in addition, we are making a selected number of invited individuals that don't have this entitlement at the level of tier structure and participation to contribute to the leadership. And I'll talk about that a bit in the next slide as well. But I wanted to leave it here just to emphasize that there is the relationship to the membership benefit is one aspect of how to participate in the program that's key. And we are hoping that as companies that are, in these the large industry levels in which they're already fully enrolled in the existing NIIMBL-led programs, that they will see the viral vector program and hope that gives them some incentive to also join our program, and if necessary to escalate their membership tier level in order to participate fully in the programs. We really welcome and would greatly benefit from that type of participation. But we know that we have a broad community, and we know that the large industry members aren't sufficient to drive all of the activities in the viral vector space that are needed. So I think I'll go on to the next slide, please, Dan, with that in mind. And I wanted to emphasize that the members that you see are the tier 1 members that are already enrolled in our viral vector program. And we have composed our steering committee with representatives from each of those tier 1 companies. And we do have FDA and NIST participating in our steering committee in advisory roles. But we also have made a selected number of invited individuals to participate in the steering committee. And this is because we recognize that we need critical mass and expertise that must be leveraged across the AAV space to fully do the right thing for the program and identify the right activities with the right technologies and the right opportunities for the community broadly. And so we've invited 2 gene therapy academicians and also have someone from one of the international advocacy organizations participating in our steering committee. And that's where we are now. We may invite a select number of additional members to the steering committee. Currently, we are composing the workstream leadership that Eric will be talking about in the analytical and process space. So that's still under development. And we plan to, again, derive critical mass, both from those member enrolled companies that are shown with the logos at the top that are entitled to participate fully. But again, plan to make invited individuals potentially from small companies, academics, consortia, and nonprofits, as well as having federal stakeholders to play advisory roles in the workstream leadership. But then, that needs to come together basically to look at the program as we envision it, bring that expertise to bear to develop the program more fully and to elaborate projects from there. So as Eric will be talking about, that we're in the process of doing this. It's going to take us a little bit more time, but we wanted to give you an overview of the structure so that you could see how you'd be able to participate. And we really do feel that it's important to take full advantage of all aspects of the community in this program to really deliver on our mission and vision. So I think that's my last slide, and I'd like to pass it over to Eric Hacherl now, who I've been delighted to and really privileged to get to work with over the last few months and partnering and helping to get this program underway. Eric, I'll pass it over to you.
Right. Thanks a lot, Tim. So as Chris talked through the NIIMBL overall structure and Tim talked through the NIIMBL-led program overall structure, you have the background for where the viral vectors manufacturing and analytics team evolved. And what I'm going to do now is walk through a little bit more of a technical overview. So we're going to go one level deeper into the program, explain how we are structuring the program specifically, and some of the feedback that we received from workshops and NIIMBL national meetings over the last several years, and really try to bring home to you what this program is going to look like, how we're going to move forward, and how you can get involved. So Dan, if you could go to the next slide. So this is a mission and vision statement, and this is specific to the viral vector program team. So different from the mission and vision slide that you saw earlier that Chris went through for the NIIMBL overall. Fundamentally, our mission has two parts. And the first part, and the most important part for us to get right, is that we create an open source or shared access platform for AAV gene therapy based vectors. And so what that means is that we intend to develop a full platform, a manufacturing process that includes drug substance and drug product, and the analytical tools to support that process, that the platform is robust, that it is affordable or economically viable, and that it can be used for tech development, for manufacturing, and for characterization. So that is the fundamental mission for our program team. As Tim mentioned, we have a secondary mission, which is focused on workforce development and workforce training. We need to be sure that we integrate this platform into existing curricula or existing training models, and also that as we go through these trainings, we can take improvement ideas that feed back from the training programs and integrate them into the platform process. And that's really critical. The overall vision, sort of the industry vision, if you will, is that the gene therapy industry is capable of serving patients across a full spectrum of need. So from prevalent indications to rare diseases and ultra rare diseases. And that industry has access to high quality viral vectors without or with reduced cost limitations and with reduced speed to market limitations. So you can go to the next slide, Dan. This slide is a little bit busy, but what I'm really trying to communicate on this slide, having talked about the mission and the vision, is that we are segmenting this program into two different work streams, process and analytics, and I will give you a lot more information on those work streams in coming slides. But fundamentally, the process work stream, as I mentioned, we are intent on developing a platform process and then looking for ways to do continuous improvement on that platform process as the industry advances. Around the analytics, we are also creating platform analytics, so tools that can be used for, primarily for process characterization, batch release, and stability monitoring, and also reference materials. And the benefits of this, we really have missed the mark if we can't reduce the individual investment cost and resource costs to develop new gene therapy programs. And that's a primary focus that we're going to have throughout this program team lifecycle. We We'll focus on improving yields for the manufacturing platforms that we deliver. We will be consistently analyzing viral vectors and the resulting clinical outcomes. We will be able to generate a lot of really valuable data through this shared platform approach. Through the data, through the experimentation, through the validation that we're able to do, we will increase confidence in viral vector quality attributes. And then ultimately, we have an aim to streamline the regulatory review process through standardization and integrating increased evidence of product quality, safety, and efficacy. So you can go to the next slide. I'm going to spend a bit of time on this slide, and this is really trying to express what the process and analytics work streams are intended to do. The information on this slide was informed through probably 2.5 years of NIIMBL efforts at NIIMBL national meetings, annual meetings, and at workshops that have been set up over the years. So this is really informed from the community, both NIIMBL member community and open community. The process work stream that we will create is going to focus on two primary endpoints. One, again, I'm going back to the platform process. So we are delivering a set of unit operations that comprise a platform. And, within that platform process, you can see 4 sub-bullets that really march through the drug substance and drug product manufacturing steps. So the HEC expression system, we are going to start with triple transfection-based upstream processing, create protocols for that processing, provide information on key starting materials, and the documentation that's needed for freedom to operate. Media and upstream processing, again, chemically defined media, but we will provide you with those key media, protocols for serum-free suspension, and process performance capability demonstrations. The downstream toolkit will include purification processes for multiple AAV serotypes, lists of affinity resins, full capsid enrichment, and process application notes across serotypes and transgenes, leading ultimately to anticipated validation studies. And then again, with fill finish, storage and stability, platform formulations, container closure, defined container closure systems, and supporting stability data and validation studies. So hopefully that gives you more of a sense for when we are successful and we've delivered this platform process, what actually is delivered to the community. And then we recognize that that platform process is not fixed in time, that continuous improvement is going to be required. And we will be looking at primarily through capacity enhancements or new technologies to expand on that platform process. So you see SF9 referenced here, and it's really reflecting a potential opportunity to increase yields through using baculovirus expression system. So we may be able to add that into the overall package. The scalable transient HEC 293 system, looking for ways to improve productivity and scalability for that base system, improved expression systems, perhaps packaging or producer cell lines can be added to our toolkit, and then downstream improvements, improved affinity ligands, improved purification processes that can be added to our toolkit. Moving across the page to the right-hand side to the analytics, the first goal is supposed to parallel what's happening on the process side, which is a platform, which is a toolkit that can be used to support batch release and stability for the base platform process that we create. And also a set of tools to allow for and enable product and process characterization. Quite a bit of data will come out of the studies that we support through the analytics pathway work stream. And we will be able to pull a lot of this data and share it with the community. And then finally, we have reference resources that we'll create. And the reference resources, you can think about them as two different types of resource. One is a physical resource, materials that we can generate. So whether this is final bulk drug substance that we can aliquot and provide to members to do interlab studies or multi-site studies, it'll be physical material that we're able to share with the community. Also, we will have documentary standards. So ed reference data that the community can access. And if you could go to the next slide. So this Gantt chart is really intended to convey how we're going to operate. We've told you what we plan to do. We've told you that we are going to create these two work streams, the analytics and the process work streams. But how are we actually going to mobilize this and generate product for the community? So the very top of this slide is the planning exercise. And frankly, this slide started back at the end of 2020 and just weighted things too far to the right-hand side when we showed 2020. But we started this overall exercise late 2020, early 2021 through NIIMBL national meetings and workshops, as I mentioned. We launched the program on October 10th. That was a very exciting date for us. And we are in the process of populating the analytics leadership team and the process leadership team. And we anticipate that will run through the end of this year and perhaps into early next year a bit. What we will rely on these leadership teams to do then is to develop the scope of work for the leadership team, to develop the key projects that need to be executed among this team and funded by NIIMBL, and then to prioritize those projects. And ultimately, the result of that will be that we will put out to the community requests for information, requests for application, and we will fund projects that, so, you know, this is represented in sort of the blue middle section and then the gray project work section. There is one case study that we're working through the system, which is a next-gen sequencing request for information that went out at the end of summer and was closed in early September. We were reviewing the information we got back from that RFI, and we intend early next year or late this year to submit an RFA for specific next-gen sequencing multi-site study, multi-lab study. And so you can see in the project work, the top bar says NGS project work, and that's the 12-month period that we anticipate funding project work specific to that NGS RFI. The remainder of the starbursts, RFI, RFAs in here are to be determined, and they will be determined by those leadership teams based on their prioritization, and then funding will follow after that. And then ultimately, there's a communication band that we need to keep very active. We do this through this webinar series. We started this last year, and this is the second of the webinars. We also will do significant communication at the NIIMBL national meetings. The next national meeting is coming up in June of 2024. And then we'll have ongoing communication, which I'll discuss on the next slide. So if you could go to the next one, Dan, I will wrap with this slide. How do you get involved? So if what we've told you about NIIMBL and the NIIMBL-led programs, and specifically the viral vector program, is really of interest to you and you want to contribute, there are multiple avenues that you can follow. As NIIMBL members, you can select viral vectors as a program or a work stream based on your tier entitlement that Tim reviewed. And you also can respond to RFIs, RFAs, RFPs to participate in program projects. We will have a survey that will go out to all attendees of this webinar, and we encourage you to use that survey. It's an anonymous survey, but we did put in an open text box. You can use that box to put in specific questions or to share relevant capabilities where you think you can contribute. And we need you in that when you fill out that survey to identify yourself and provide contact information, because it is anonymous. So we won't know who you are unless you tell us. We have the NIIMBL website and NIIMBL portal. The NIIMBL website is an open access website, and you can sign up for NIIMBL's general need to know newsletter. So major announcements for the viral vector program will come out in that newsletter. Also, we have a specific viral vector mailing list that is open to all. The link is provided here to sign up for that mailing list. For NIIMBL members, you can choose to follow the viral vector program, and Angie will explain that to you in a minute. And then finally, community workshops. We do envision that we will continue to hold periodic member and or public workshops to inform on our progress and to get feedback on areas that we need to focus on. These could be related to either the overall viral vector program or to one of the specific work streams, process or analytical, or it could be focused on a specific technology or a specific project that we're working on. And that's it. So I'll hand it over to Angie, and she can wrap up and then lead to questions. Angie?
Thank you. I'm going to show a slide that does show you how to follow the programs that Eric was just describing. So as a reminder, if you're a NIIMBL member, you can choose to follow projects and programs through our portal, and you can receive updates to your inbox. To do that, you first need to be logged into the portal. So you're going to do that in this picture up here in the right-hand corner. Then you can search the projects and the programs from the toolbar. And then when you're logged in, you'll see both the overview and the updates tab. If you're not logged into the system, you're only going to get the overview. So just keep that in mind when you log in and you can't see the updates tab. And then to follow the program, you're going to click the teal follow button on the right. And just to let you know, if you choose to follow a bunch of different projects, you're only going to get one weekly email with the updates. Dan, you want to go to the next slide? Okay, so now we're going to take some questions. We have a few minutes for questions. Before I start asking the questions, we've received quite a few chats through the, quite a few questions through the chat, so thank you for that. I'm going to try to paraphrase them together because we have some common themes. But if we don't get to your question, please feel free to reach out to us after the webinar at the communications email you see below. And so let's start with a question for Tim. Tim, can you talk about the relationship between the NIIMBL-led viral vector program and the open project calls? And a follow-up to that is everything that is viral vector related or AAV related expected to happen within the NIIMBL-led program?
Thanks for that. It's a great question. Appreciate it. The short answer is no. Everything that's AAV related or viral vector related should not be expected to happen in this new NIIMBL-led program. In fact, there have been many projects that have already been funded and conducted through the open project call process, and we expect that to continue. So I really see the open call, or as we call it, PC program, and the NIIMBL-led programs to be complementary. We really need both of these in order for NIIMBL to succeed, and they play a bit of different roles. And I think, as I mentioned, the open calls, the projects and ideas are basically investigator initiated. We articulate needs in particular areas as part of the open calls, and then people respond saying, I have an idea that can fulfill this need. And this I really look at as a key innovation engine for NIIMBL and for the ecosystem as a whole. Many of these perhaps are novel ideas that or applications into the AAV space in our case, where something may not have been tried for AAV that really has an opportunity to step forward and be brought to a level of interest for integration into AAV-based manufacturing and analytics. And the NIIMBL-led program would basically be sort of the complement to that, where we'd be saying, let's help to demonstrate how the full reduction to practice of this, what the implications of that would be, and essentially to demonstrate it in the context of how manufacturers would use the processor analytical tools and to demonstrate the positive effect of them. So I think the roles are different, but they're very complementary. And and both are important. And our intention would probably be to keep pushing for the open project calls to generate new ideas and to keep watching those on the NIIMBL-led program side for maturation and pull into the NIIMBL-led programs. I have to admit that we probably aren't fully organized yet to do a great job of that, but it's on our radar as something that's really important to make sure that we leverage and relate the NIIMBL-led program to the open project calls to take full advantage of the whole ecosystem. So thanks very much for that question.
Okay, great. Okay, Eric, I'm not going to leave you out. How will the question is, how will I hear about RFIs, RFAs, and RFPs? Maybe you could explain that a little bit.
Sure. I'll use the NGS RFI as an example. So we will provide access to RFIs, RFAs, and RFPs, both to the NIIMBL community and also through the different channels that I mentioned in my How Do I Get Involved slide. So RFIs, for example, will go out to our mailing list. They will be posted on our viral vector program page. And so you have, and we'll use social media also to communicate that the RFIs have gone out. So there are multiple different channels to hear about RFIs, and we ask, we will provide information on what specifically we are requesting, what the due dates are, and how to submit applications. And we encourage you to apply that way.
Okay. Tim, you talked about making invitations to program leadership, both the steering committee and the work streams, to add critical mass. What can you say about how you're going to go about doing that? What are you looking for in the way of nominations? Are you asking people to raise their hand? What's our process going to be?
Yeah, thanks for that. I actually didn't dwell on this, Angie, in relation to what you said I might as I talked about it, but this was actually a bit of a challenge for us in relation to the status quo of the NIIMBL-led programs. When the first NIIMBL-led programs were launched, which you may know are process intensification and the big data program, this viral vector being the third, those first two were heavily focused on large industry, large biopharma manufacturing capabilities and needs and how to advance those, perhaps in different ways, but in particular, the PI program, as we call it, is focused in the protein therapeutic space where there's been a long history. And so we actually had, I think, 13 program participating companies or some number, 13 or 14 large industry participants in that program. There's a huge amount of embedded expertise there in protein biopharmaceutical manufacturing that was really driving the program. So enormous critical mass already available. In our case, we were recognizing the maturation of the industry. It's more like 20 years ago for the mAb industry, maybe even longer. And the amount of expertise that's embedded in the small companies and in the academic community, the weight of that and its level of expertise and understanding of what the needs are just cannot be denied, and it can't be missed out. So we recognized we needed to do something structurally to enable that. So this notion of an invitation came up as a way to expand the critical mass without actually undermining the tier benefit structure that NIIMBL has created. And so we worked this up. And so there are some key parameters of these invitations. One, I need to emphasize that they're invitations to individuals, not to organizations. So while everybody represents an organization, the organization doesn't actually receive any rights that go along with the invitation to the individual that's participating. So the rights actually remain tied to those member benefits that I previously described. But the purpose was to ensure that we have that specific and relevant expertise to guide the technical program. And so we wanted to make sure we had critical mass, but we also can't have the steering committee or the workstream leadership teams be so large as to be unwieldy. So we need the right kind of balance there. And we want, in the purpose of fairness, we also need to ensure that the voice of the tier entitled participants in the leadership remains strong. So we basically place some constraints on how many invitations we could make, so that the voice of those that have basically bought in at the highest level to participate in the program have a leadership voice that doesn't get overly diluted by the participation of the invitees. So the process that we've been using to identify and make these invitations, we really have been looking for people that have clearly demonstrated the expertise in the AAV space, process and analytics, have shown an interest in the NIIMBL-led program through our participation in workshops, or not only our NIIMBL workshops, but other workshops, conferences, and really a passion for the mission of this program. And one thing that we're sensitive to is that we want people that are providing leadership to this NIIMBL-led program at the steering or the workstream leadership levels, to really embrace the NIIMBL mission and the program mission on behalf of the entire community and ecosystem, and not simply be trying to come in there to look out for their own interests per se. So that was sort of important to consider. And with that in mind, I have been identifying individuals and then bringing those to our steering committee with the tier entitled members and asking them for approval for each of the invitations. So that's the process that we've been using. So we don't actually need people to self-nominate per se. Of course, we're interested in people that are interested, but we don't actually have a formal nomination process. We have a pretty good network that we're trying to leverage here, and then getting the approval of the steering committee. Thanks for the question. Sorry for the long answer.
Okay. Eric, is NIIMBL open to considering other vector modalities such as LNP or lentivirus?
Yeah, thanks, Angie. What we have constructed for this NIIMBL-led program is based on feedback from the community over the last couple of years, and really has focused on AAV as the most advanced of the modalities and the most likely to demonstrate quick results, and particularly focusing on the ultra rare disease community, where getting an open access platform pulled together quickly is really of utmost importance. We are focusing on AAV for now, and it does not mean that we won't focus on other modalities in the future, but for now we are exclusively focused on AAV. And I do want to answer one of the chat questions about this Bespoke Gene Therapy Consortium in this regard. We are contributing this process as a NIIMBL is a member of BSGT, Bespoke Gene Therapy Consortium, and we are contributing this process to BGTC to be used by that program also. So focus on well-developed existing technologies initially that can be deployed in an open source format. Thanks, Angie.
Perfect. Thanks for putting those together. Okay, one more, Tim. I love this question because we hear this a lot. Will the NIIMBL viral vector platform introduce a new cell line, new plasmids, new filters, etc., or does the NIIMBL platform use existing technologies that are possibly not license free?
That is a great question. We do get it a lot, and it is a challenging one to answer. Probably by intent, yes and yes. So as Eric mentioned, we are likely, for expedience, to focus on existing technologies that are fully synthesized and made available as a sort of core and urgent first step. But we also don't want the platform technology to be dead on arrival, so to speak. We don't want it to be old technology by definition. And there's also a question in the chat about cost of goods. This is a significant access issue, and cost is very important. So we do anticipate, in parallel with working on pushing out core platform technology, we do anticipate enabling and trying to draw upon advances in the technology that will improve process and analytical performance, and then integrating those into the platform as time goes on.
Okay. Okay. I think that will call the questions there. And Dan, can you go to the next slide, please? Okay. As a reminder, you'll be sent a survey soon after the webinar. And as Eric stated, there will be a question where you can self-identify and let us know if you have interest in the program. You do need to self-identify because it is anonymous and we won't be able to track you down if you don't tell us who you are. And then our final webinar in this series will be held after the Thanksgiving holiday, where John Balchunas will be talking about expanding talent pipelines. And then the next slide is just a thank you for joining us. And again, if you have any other questions, let us know, and enjoy the rest of your day. Thank you.